Hello everyone, welcome to the channel. My name is Kevin and I would like to share my research topic and my progress of this project in this video. So the topic of this project is called the effect on development of fovea in green anole by causing a mutation on Foveal Hypoplasia related gene PAX6. In other words, what are the changes to the phenotype of the fovea or even the eye development after causing this mutation? So first of all, what is a fovea? Fovea is a depression at pretty much the center of the retina in our eyes, which are shown in this picture. Here, this is the fovea. And compared to the normal retina layers, fovea layers have high density of cone photoreceptors that are responsible for highest visual acuity and identification of colors. So for people with normal visual acuity, for instance, if here there's an object over here and you're looking at this object through your eyes, the light of this object will transfer through this lens and then finally congregate on the fovea. Now, what happened to the people with Foveal Hypoplasia? So then what is a Foveal Hypoplasia? Foveal Hypoplasia is a genetic disorder with features overlapping aniridia, hereditary keratitis, ocular albinism, and iris anomalies. So here in this picture, this is a patient with aniridia, and it is compared to a people with normal eyes. So as you can see, this patient do not have iris, and the pupil is abnormally large. And for hereditary keratitis, this simply means you have inflammation of the cornea. And for ocular albinism, the symptoms are something like involuntary eye movements, which is the nystagmus here. They also have that their eyes that it cannot look in the same direction, and you become really sensitive to light. So this genetic disorder is mainly caused by the mutation of this PAX6 gene. And the PAX6 gene is greatly expressed during the development of the eyes. The mutation of the gene is autosomal dominant in an affected individual. So in this picture, we have a affected father with a single allele mutated allele. So when he mates with this unaffected mother, after the allele starting pairing and pairing, the second generation will have 4 possibility of the chromosome pairing outcomes. As you can see, half of the second generation will be affected. So there are several reasons I chose green anole as the model organism to study. So the first one is a lot of previous eye studies were done on animals such as mice, zebrafish. However, these model organisms do not have a fovea. So then people start to study fovea on animals such as primates, but primates have really low reproduction rate and they're not easy to manipulate. What is more important, they're really expensive compared to mice, zebrafish, and green anole. So we have to come up with a better model organism to study fovea. So compared to primates, green anole is rather cheap. Its reproduction rate is pretty fast, and they're really easy to manipulate because they're small. Second of all, we know the genome sequence of the green anole, and it's been unveiled in 2011. So the good thing about this is that because we know the genome sequence, this will make genetic study much more easier. The third reason is that green anole have similar central fovea structure and development compared to humans. And finally, recent studies have been proved that we can manipulate CRISPR-Cas9 gene editing strategy in green anole. So now let's talk about eye development from blastula to formation of optic cup. In human, after the process of gastrulation from blastula, usually 3 germ layers are formed. These 3 layers are ectoderm, mesoderm, and endoderm. The ectoderm will form the epidermal layer of the skin and the nervous system, including your eyes and nose. The mesoderm will form muscles, bones, kidneys, blood, gonads, and connective tissues. And finally, the endoderm will form your guts, your liver, and lungs. So here, we focus on the neural tube that is still part of the ectoderm in this stage. In this figure, the neural plate then starts to invaginate, and then finally, it's separated from the ectoderm. This neural tube will later on develop into your brain and spinal cord. Our eyes are also developed from this tube, and it's usually developed at around the area of brain. The development of eyes is first discovered at around day 22. And here, we cut the head of a fetus through this dashed line, and you look through the head in this direction. At around day 22, the 2 optic grooves will start to elongate bilaterally and start to approach the surface ectoderm areas. At day 28, after the optic vesicle reaches the ectoderm, it induces the formation of the lens and form a lens placode. At this surface ectoderm area, you can see they are thicker than the other surface ectoderm area. Then this lens placode start to invaginate inwards, just like how neural plate invaginate inwards to form the neural tube. And finally, the lens vesicle will form at around week 6. And at week 6, it is called optic cup because here you can see a shape like cup that form. And this cup, it's related to our main characters in the story because there are 2 layers. The inner layers here will later on form the retina, and the outer layer will later on form the retinal pigment epithelium. So the process from the blastula to the formation of the optic cup usually happens between the embryonic stage 1 to 4 in green anole when the egg is still not laid. Anoles have 19 embryonic stages, and the eggs are usually laid at about embryonic stage 4, or probably right after embryonic stage 4. And they hatch within 7 weeks if the correct amount of heat and humidity are maintained. And starting from embryonic stage 5, we're able to observe the development of the retinal layers. So before we talk about specific development of the retinal and foveal layers, let's first take a closer look at what those layers look like and what functions they have. So the retinal layer structures in vertebrates are pretty similar. And here, this is a picture of a human adult retinal layer. And it looks pretty complicated, but don't worry, we have a simplified version in the next slide. And as you can see, we have many layers in our retina, and normally the light signals will be transmitted from the inner retinal layers to the outer retinal layers, basically in this direction, all the way to the bottom. And the neural signals are right opposite to the direction of the light. When the neural signals approach and reaches the top of the retina, they will be transmitted by this optic nerve at the surface of the retina here. And these optic nerves will all congregate at this optic head or optic disc, and then the optic nerve fibers will be transmit the signal to the brain. So below the nerve fiber layer, the ganglion cell layer are basically made up of ganglion cell bodies. So you can see we have ganglion cell here. So these cells are really just responsible for transmitting the signals to the brain through the nerve fibers that extended from the cell body and receiving the signals from the cells that connected to them in the inner nucleus. In this inner nucleus, these cells are amacrine cells and bipolar cells. So the amacrine cells are mainly responsible for modulating the signals from bipolar cells before transmitting them to the ganglion cells, while the bipolar cell receive visual input from the photoreceptors and the horizontal cells below. Horizontal cell and the photoreceptor cells below. And like the amacrine cells, the horizontal cells help modulating the signals from the photoreceptors and then transmit them to the bipolar cells. So the amacrine cells and the horizontal cells are basically just the modulator of the neural signals. And then the photoreceptors in the outer nuclear layer, the inner nuclear, the inner segments and the outer segments are playing a really important role in building a normal vision. So there are two types of photoreceptors. They're labeled here. They are cone and rods. Rods are responsible for what is called scotopic vision. Scotopic vision. And which simply means just promoting vision under low light level conditions. They do not usually function during daytime because during daytime you usually are in the conditions of high light levels. And cones, on the other hand, are responsible for what is called photopic vision. So photopic vision, which simply means the vision under normal or high light level conditions. And they allow for greater spatial acuity and they can mediate colors. In human eyes, there are three types of cone cells. There are three types of cone cells. These three types are specific for short, medium, and long wavelength of light. You know, there's an additional cone cells that can help detecting ultraviolet. So anoles have 4 cone cells, and the additional one can detect the UV light. This means that anole can see a wider wavelength of light compared to us. Then below the outer segments, we have retina pigment epithelium. The retina pigment epithelium takes care of the photoreceptors and the retina. They can transmit nutrients from the choroid to the photoreceptors and receive waste from the outer retinal layers. They're also important for visual cycle, renewal of the outer segment tips, and absorbing lights that may damage the retinal layer. And finally, the choroid is just made up of blood vessels that can supply nutrients and oxygen to the retina. So compared to the normal retinal layers, the fovea is much different. Except for the photoreceptor layers, all the upper layers are gone. So where do they go? You will know the answer when we talk about the fovea development in the next section. And more importantly, fovea only consists of one type of photoreceptors, which is just the cone. It has much denser cones than other retinal layers, and hence the fovea is responsible for the highest visual acuity and precise color vision. There is one prominent difference between human and anole fovea layers is that the anole do not really have outer nuclear layer. As you can see in the central fovea region, there's no photoreceptor cell body here. So why this happens? The reason for this is still under discussion, but one of the guesses is that anoles have higher density of cones compared to human. Another thing I need to highlight about the anoles is that besides central fovea, they also have an addition fovea called temporal fovea. The function of the temporal fovea is to mediate the binocular vision of the lizard. And in our project, we mainly focus on the central instead of the temporal fovea. So make sure you don't confuse about which is which. In this section, we're going to talk about fovea development in anole. First, let us talk about the sectors of anole retina. Usually, the retina of the vertebrates is developed by two axes, the vertical axis and the horizontal axis. The horizontal axis differentiates the retina into superior, and inferior sectors, while the vertical axis differentiates the retina to temporal and nasal sectors. And of course, the nasal sector is the one that is closer to the nose, and the superior sector is the one that is pointing upward. There is a conventional rule of retina development in most vertebrates. The development of the retina begins within the central retina, then it progresses through the temporal retina and concludes in the nasal retina. This phenomenon is also shown in anole's retina development because later you will see that all the processes such as cell differentiation and retinal lamination all starts at the future central and temporal fovea regions and ends at the nasal regions. So we previously mentioned that the optic cup is formed at around embryonic stage 4, and that is when the retina start to develop. At embryonic stage 5, the egg is just laid by the mother, and here we can see that the retina is still filled with what is called the mitotically active cells, one layer of mitotically active cells. And these cells have the potential to further develop and differentiate into neural cells that we discussed in the previous sections. These cells are, ganglion cells, bipolar cells, endocrine cells, photoreceptors. And here is this figure A. This figure A shows the retina of the anole at embryonic stage 5, and we use this dashed line to discriminate the nasal and the temporal sectors of the retina. So the left part is nasal and the right part is temporal. The sections labeled B, C, D, E, and F in figure A have been zoomed in. The figure D shows the future central fovea or the central retina, while these figure F shows the future temporal fovea region. As you can see, these two regions are obviously thicker than other areas of the retina. And although no obvious lamination is observed in stage 5, many neural cells have been discovered, especially at the regions of central fovea, central retina, and temporal fovea regions. These cells are ganglion cells, horizontal cells, and photoreceptor cells. At embryonic stage 6, the first observation of lamination was discovered as IPL here and OPL, also shown in future temporal fovea region. And also at this stage, the amacrine cells are first discovered at the central retina, which is here. You can see a really clear discrimination of a single layer of amacrine cell here. Other existing neural cells are also keep developing and shows up in one other peripheral areas of the retina. The other type of neural cells, the bipolar cells, are not detected until the stage 9 to 10. So from stage 9 and 10, we can clearly discover that inner nuclear layer has been divided into two sectors, which we mentioned in the previous slide. So as you can see in this figure G, there are clearly two sectors in this inner nuclear layer. So the upper layer or the inner layer is the amacrine cells layer, and this outer nuclear layer is filled with bipolar cells. At the same time, the photoreceptor cells in the anole have developed into a 1 to 2 cell thick layer. As you can see in figure F, below this outer plexiform layer, which is pink here, there's a dark layer, which is the outer nuclear layer, and they're about 1 to 2 cells thick. From embryonic stage 9 to embryonic stage 14, the eye undergoes a process called ocular elongation. This process will elongate the retina and can decrease the cell density of each layer, including O and L. And before the start of the fovea formation, O and L stays in about 1 cell thickness, even though more cells are growing during this time. And what is more, inner segment and outer segment of the photoreceptors are not shown from stage 9 to 13. Then at embryonic stage 16, the formation of the fovea depression becomes slightly significant. What is more, the outer nuclear layer developed from 1 cell thickness to approximately 4 cell thickness in this figure B here. More and more photoreceptors start to develop, and because the central fovea needs to have really high density of cones, the inner retinal layers need to undergo a displacement process to provide more space for the cones to develop and grow. So starting from embryonic stage 16, the GCL, IPL, INL, OPL, and even the ONL, they all start to develop and displace to the peripheral foveal regions. The photoreceptors become mature as the inner segment and outer segment extend from the outer nuclear layer vigorously from embryonic stage 16 all the way to the hatchling stage. And due to the high density of the cones, the displaced cell layers also have high density of inner layer cells that connect into those cones in the fovea. So as you can see, at around the hatchling stage, you can see these two parts, the peripheral foveal regions, are much thicker than other regions, such as the nasal retinal regions. After the egg is hatched, it still takes about 3 months for the fovea to be fully mature. And within these 3 months, the foveal photoreceptors keep increasing their density to perfect the function of the central fovea.